Hey everyone, thanks for joining for our final panel of the day. I'm Julie Nania and I have the pleasure of moderating our panel on sage-grouse habitat restoration and management. Yeah, we can share for that. Today we've got three of our MEM candidates with us. We've got Ema Muslic, Patrick Kenney, and Sophia Reggiani. And I'm going to hand it over to them to introduce this panel and get things started.
Right. So the sagebrush steppe ecosystem is a very unique ecosystem that comprises about 2.5 million acres in the Upper Gunnison Basin alone. It comprises of grasses, forbs, sagebrush, and other shrubs, as well as 2% of that 2.5 million is designated wet meadows. The headwaters of the Colorado River system start in sagebrush steppe, as well as multiple other large river systems. And it provides ecosystem services, including but not limited to nutrient and sediment retention, snowfall and rainfall capture. And lastly, it's important for ranchers, livestock, the survival of the Gunnison sage-grouse and greater sage-grouse, and wild game forage. More than 170 vertebrates and tens of thousands of invertebrates call the sagebrush steppe home.
Thanks to our very own provost, Jess Young, the Gunnison sage-grouse is considered threatened under the Endangered Species Act. This bird lives mostly in Colorado with a small population in southeastern Utah. Gunnison sage-grouse co-evolved with the sagebrush and is highly adapted to it. It depends on sagebrush leaves solely during the wintertime. In summer months, the sagebrush is reliant on mesoforbs and insects found in nearby wet meadows, riparian areas, and irrigated fields. Riparian areas are wet meadows are particularly important for Gunnison sage-grouse brood rearing, as the sage-grouse chicks depend on the high caloric and protein-rich diet found in wet meadows, just so that they can grow big enough, quickly enough, so that they can fly away from predators. Because this species is so dependent on the Gunnison sagebrush and a particular mosaic of habitat, it's highly at risk of becoming endangered if we don't manage and care for these habitats carefully.
As climate change is predicting warmer temperatures and less water on the land, it is important to protect these sagebrush habitats in an already arid ecosystem, with wet meadows being an oasis in this high desert. Wet meadows are historical land use practices, grazing, invasive grasses, and in conjunction with climate change is going to exacerbate these changes. And the existence of these wet meadows, sagebrush, and the organisms that live there. Wet meadows and sagebrush restoration requires long-term post-restoration management and monitoring, which is crucial to inform future land management practices. With that, we're going to head into Emma's presentation. The clicker will work.
So we're going to have Ema first, and throughout this process, same as the other presentations, we're going to have an opportunity to ask each panelist questions at the end of theirs, and then to ask the panel as a whole questions after everyone has had a chance to present.
All right, hello everybody. My name is Ema Muslic. I'm an MEM student as well as an MSE student. Today I'm going to talk to you guys about my MSc thesis work. Hold on. These are the pictures we were trying to show you guys when we were doing the intro. Very beautiful. Okay. Ooh, there we go. Okay, so my thesis research is regarding herbicide effects on cheatgrass and native plant species in the Upper Gunnison Basin. So just to give you guys a brief overview of cheatgrass or Bromus ductorum, it's an invasive annual grass. What that means is the fact that it is able to germinate very quickly in the spring and winter months, and actually create seedlings under the snowpack while other plants are not even at that point. They're not even developing. And so once the summer comes around, those plants, cheatgrass plants, are able to mature, drop seeds, and dry out very quickly in the middle of the summer. Cheatgrass is spreading across the Intermountain West very quickly at a rate of about 2,000 kilometers squared per year. This is negatively impacting both the sage-grouse habitat as well as native grass habitats. The reason why is because it's taking up all these resources and otherwise habitat that would have been used for their benefit, such as forage, nesting, breeding, and so forth. On top of that, cheatgrass is a very big threat to the fact that we're having more wildfires more frequently. Cheatgrass, like I said, dries up very quickly and is one of those fine fuels that once lightning strikes or a campfire were to go astray, it is that first plant that's going to spread the wildfires even farther and faster. One thing that I'd like to point out to people is that this doesn't just affect wildlife habitat, but that also affects our livelihoods and what we love doing, which could be hunting, hiking, bird watching, etc. So one of the management techniques that is used to get rid of or try and mitigate cheatgrass is through herbicide application. That's what we're going to talk about today. The two herbicides that we used in my experiment are imazapic and indaziflam. Imazapic, which is the most widely used and has been used across the last couple of decades, is a pre and post-emergent herbicide, which means that once it's sprayed onto a plant, it effectively prevents that new plant from producing more seed, kills it, and also kills the seeds that it has already dropped that season. It is known to be used every couple of years after application. You usually have to reapply it every 3 years. For indaziflam, which is a newer, just recently approved herbicide to be used on public lands, this is a pre-emergent herbicide. So it's mostly targeting the seeds in the seed soil bank. So cheatgrass seeds can be viable in the soil for up to 5 years. Is what's been found. So this is kind of promising for indaziflam because it's not only getting that first season of application, but it's also getting into the seed bank, getting seeds from 2, 3, up to 5 years earlier. So one of the questions we wanted to know was whether or not a combination of the two herbicides applied at once could be an effective way of mitigating cheatgrass without having to apply herbicides as often as we are now. So my first objective of this experiment was to assess the effectiveness of imazapic, indaziflam, or the combination on the reemergence of cheatgrass. And then the second was to measure any of those effects of the treatments on native plant species. My study area is in Dillon Gulch, which is about 20 miles west of Gunnison in the Dillon Pinnacles region. It's about 2 miles north of the Dillon Pinnacles trailhead. So it doesn't see too much foot traffic or recreation traffic, which is great for an experimental plot to be had. It's an east-facing sagebrush steppe habitat. It sees a very moderate cheatgrass invasion. So overall, that whole east-facing slope, there's about 20 to 25% cheatgrass invasions across it. It has a wide range of cheatgrass invasions across elevation and slope as well. So as you can see, the purple dots here where I actually had my study sites put are across a zigzag pattern across that elevation type. And the reason I did that was because I didn't really want to do it in one specific elevation and create any kind of bias with that. And then, of course, like I said, low foot traffic. So for the experimental design, this is a 3-year experiment. It's still ongoing. I have my last field collections this summer. But 2 summers ago, before I even started any classes, I was able to go ahead and hike out to these study sites and do a pre-treatment survey of the plant composition. So just seeing what plant species are even out there along with the cheatgrass. And then last summer, I did my first post-treatment survey. And then again, this upcoming summer, I'll be doing the last one. So this experiment has 10 blocks, which are 100 meters apart from each other. These are, like I said, across different elevation ranges. And within those 10 blocks are 4 plots with a random treatment assigned to it. So that could be your control, that could be indaziflam, imazapic, or the mixture of the two. And they were put about 6 meters apart from each other to prevent any intermingling of the treatments. All in all, there were 40 plots across 70 acres. On top of that, the plots where the treatments were actually done were put in a 1 meter by 1 meter grid, which is where the plant composition surveys were done. I'll give you guys a little overview of that method in the next slide. And then for the actual treatment, we did a 3 meter radius. So on that actual plot area, as well as a little bit outside of it, just in case there was any wind dispersion or water dispersion of seeds coming into that survey area. The sampling method that I used was the grid point intercept method, very similar to the line point intercept method, but just kind of put into a very smaller area. The line point intercept, it's like 50 meters. This one is just a 1 meter by 1 meter plot. Within that frame are 100 individual crosshairs where I was able to take a survey flag with a thin metal stake, drop that down on each crosshair, and record any type of species of plant that was touching the survey stake. And then that was recorded into a data frame, and we got a count of the different kinds of species per plot. On top of that, I also took aerial photographs of the plots to get a visual composition over time. I'm not going to show those with you guys today just because of time constraints, but if you have questions, let me know. And then lastly, for sampling, we did some seed bank viability sampling. So like I said, seeds can be in the soil, the surface soil, up to 5 years. So we wanted to see that first year post-treatment, whether or not some cheatgrass seeds would come back from those treatment areas. So we were able to take 4 samples, surface soil samples from each of the plots, totaling 160 soil samples. Those were then stored in a cool area, dried out, and then when they were ready, put into the greenhouse for germination. So this one here on the right is one of the cheatgrass plants that came from one of the soil samples. And once you're able to identify that it is cheatgrass, you again do count data, and we'll be doing the same type of analysis I did with my cover data. So going back to the plots and the cover data, the 3 main things I wanted to look at was cheatgrass cover over time and treatment type, forbs cover over time and treatment type, as well as native grasses cover. So these are very preliminary results. We still have another season, but for now, this is what it looks like 1 year post-treatment. So as you can see here on the left, the lighter colored boxes are going to be pre-treatment. So what each treatment type had for cheatgrass cover, and then the dark red is going to be what the cheatgrass cover looked like afterwards. So of course the control, not a lot of difference. And then as you can see, the 3 asterisks show significant difference. So there was a significant decrease in cheatgrass cover across all treatment types, most notably in the mixed and the imazapic. It was essentially eradicated from some of them. We'll see what it looks like this upcoming summer. Then for forbs cover, I thought this one was pretty interesting. So the mixed and the imazapic treatments showed a significant reduction in native forbs cover, which is not what you want to be seeing from the herbicides. Although indaziflams was not a significant reduction, so that's pretty promising. And then lastly, for native grass cover, it decreased across all treatment types, including the control. I think that this has to do something with environmental factors such as moisture or precipitation amount that was had across the years. As you guys can probably remember, in 2023, we had a really awesome snowpack and precipitation, whereas in 24, it was a little bit more reduced. So I believe that that probably points to it. Then for the preliminary greenhouse count data, you can see that a lot of our soil samples had a 0 amount of cheatgrass come out. But those that did have cheatgrass emerge, there was no significant difference between treatment types. To get a better visualization of just the total amount of cheatgrass that reemerged in the soil samples, this is pretty on par to what you just saw in the last slide. But basically, the control had the most cheatgrass come back, which is expected. And then our mixed and Rejuvera had the lowest amount of cheatgrass reemergence in the soil samples. And then Plateau had a pretty big variation. So my next steps are going to be to this summer go ahead and continue my collections for plant cover data, plot photos, and seed bank samples. And then lastly, continue some more analysis with generalized linear models, specifically on the top native species that were observed. I'd like to say thank you, everybody, for joining and listening today. And also thank you to everybody that has brought me to this point so far. My committee members, Dr. Coop, Bridget, Petar, and Jared, as well as AJ, my undergrad assistant. She helped me lug back a bunch of soil samples that were like super heavy. So thank you so much. But yeah, thank you guys.
So while we're waiting for some more questions to load here, I'm curious, I know you're still in progress here, but it seems like you've got some direction for maybe some preliminary recommendations. What might those be?
Well, I would say that since indazaplam is newer and it's just been approved for more widespread use across public lands, I think we should be looking into it as maybe a supplement to other types of cheatgrass mitigation. So say prescribed fire, mechanical pulling, that kind of stuff. So kind of use it as a way to prevent the cheatgrass that we've already treated with other mechanical or fire. Use that as a way to prevent it from coming back up as early as it usually does.
Right on. And coming from the crowd, what ratio were the herbicides mixed at and how did you determine that?
So all of the herbicides were at 8 ounces per acre. That was by recommendation of the cheatgrass coordinator, who was also on my committee, Petar, at the time. He believed that that was going to be the best possible application for rangelands and areas that don't see as much recreational use.
And I think we actually have time for one more. So what was the most difficult aspect of your field work aside from having to make your field tech haul tons and tons of soil?
I think just like planning out the logistics. I think at first I was like, oh, I can do this all on my own. I can carry out this whole grid frame and a mallet and all these materials. And so it ended up taking like twice as long as I had hoped it to be the first season. And then once I got an undergrad, it was like, oh, this is a lot easier. So maybe just planning out the logistics better and having realistic expectations with that.
Right on. Well, let's hear for Emma and undergraduate assistants. All right. So next up, we've got Patrick Kenney. Come on up.
So my project was wet meadows restoration for Gunnison sage-grouse. This project came about after years of groundwork laid by Jenny Nearing and Carrie Aloya, by wildlife biologist and wetland biologist and owners of a small ecosystem management company in the San Luis Valley called Wetland Dynamics. So restoration projects take a long time to build trust and nourish relationships with stakeholders. Landowners often need to be educated about the benefits of restorative land management practices and convinced that it's a worthwhile investment. So after many years of laying this groundwork, Wetland Dynamics, in partnership with Ducks Unlimited, ended up receiving funding through a Fish and Wildlife Service Bill Sage grant to improve Gunnison sage-grouse habitat in Poncha Pass, Colorado. These habitat improvements will also benefit endangered or threatened Rio Grande suckers and chubs, as well as many other species that depend on wet meadows and riparian areas for food and water during the summer months. Wetland Dynamics is acting as the project coordinator, coordinating these improvements, and the plan is to utilize low-tech process-based restoration to repair some of the degradation caused by land use choices in the San Luis Creek drainage basin. So there's a small satellite population of Gunnison sage-grouse. This purple box over here that exists on top of Poncha Pass near Saguache County, Colorado. And there's only about 15, estimated to be 15 individuals. And these Gunnison sage-grouse are dependent on large areas of sagebrush for their survival and a mosaic of wet meadows nearby. So before human intervention, snowmelt and runoff would flow across alluvial fans and sheet flows, which would allow the water to spread out and soak into the landscape, irrigating a wide swath of land. Modern humans have caused many disturbances to watersheds through activities such as building roads, highways, irrigation canals, and fences. These changes to the landscape often interfere with hydrologic processes and have many unintended consequences. This picture is from the project site. This is the Alder Creek after it passes under the highway. It's been artificially channelized to flow through a culvert. The effect of this has been to dry out the surrounding landscapes. So in addition to land degradation, climate change has also threatened a lot of these habitats across the arid West. Over the past 2 decades, the southwestern United States has experienced the worst drought in 800 years. According to the EPA's 2006 Wadeable Streams Assessment, 44% of riparian areas in the United States have high rates of disturbance and contraction, and 49% of ecosystems have poor or fair riparian vegetative cover. So it's critical that we protect and enhance this wet meadow and riparian areas that many species depend on. For many years, restoration professionals used form-based restoration, which seeks to force a stream channel or wetlands into a particular form. This is often very expensive and takes a lot of outside inputs and energy. Oftentimes these structures end up failing because they don't factor in the dynamic and evolving nature of watersheds. More recently, restoration professionals have been using low-tech process-based restoration. This type of restoration seeks to use local native materials and use the power of water and sediment in natural heavy flow events to do the work of the restoration. The idea is to restore the natural processes that support these evolving and dynamic landscapes. Some examples of low-tech process-based restoration are simple hand-built rock structures called Z-dike structures, one-rock dams, rock rundowns, Zuni bowls, and media lunas are examples of these. In this picture is taken from a project that was done in the Gunnison County on On the top left, you can see people installing a media luna. This has the effect of spreading the water out and in turn acting like natural sheet flows. Some other examples of process-based restoration are beaver dam analogs and post-assisted log structures. Purpose of these structures are to slow down water. They create pools that act as habitat for fish and birds and other wildlife, and increase floodplain connectivity. So because resources are limited and the initial installation of these structures is very labor intensive, restoration efforts must be carefully planned. Sites within a drainage basin need to be selected on their capacity to cause the greatest impact with the resources available. Last summer, I had the privilege to shadow Sean Connor from Biologic as he hiked around the project site, reading the geomorphic context and the history of the landscape and strategically selecting where to place these structures. During the planning phase, we met with a lot of local landowners and ranchers to gauge their willingness to allow these structures to be installed on their properties. As many of you know, a lot of ranchers and farmers aren't big fans of beavers, so a lot of them wouldn't want to encourage beavers moving back into their property. Interestingly, while we were out walking the property with a longtime landowner, he told us a story about how his father used to use the beavers to help irrigate his fields. Unlike other ranchers, instead of shooting the beavers, his father would allow them to make their beaver dams, and then when they needed the water further downstream, they would blow up the dams with dynamite to allow the water flow, being careful not to hurt the beavers. So after gathering the information we needed to decide where to put these structures and what structures to put, we made a plan, which was to use volunteers to install these structures in the summer of 2024. According to that plan, we're going to install one rock dams and rock rundown structures in the ephemeral streams, which slows down runoff and allows the sediment and nutrients to drop out of the water column, and it also captures seeds. So yeah, this reduces erosion and allows the water to spread out and soak into the surrounding landscape to be used later in the season by plants. In the perennial streams, we're going to be installing beaver dam analogs and post-assisted log structures to increase stream channel structure and create pools that act as habitat. And then these stream channels also induce braiding, which spreads the water out and wetting more of the floodplain and increasing groundwater storage. Under ideal conditions, beaver dam analogs will encourage beavers to move in and take over this process. So for my project, in addition to some of the other things I did, I will help direct volunteers to build these structures in the stream channels around the San Luis Creek drainage. And then to track the efficacy of these structures, I've been establishing a pre and post monitoring restoration study to track vegetation changes over time. So in the summer of 2024, I collected baseline data, including line point vegetation surveys using the BLM's AIMS protocols. Additional vegetation surveys will be conducted this coming summer. We also, in 2024, with Dr. Crosley's help, flew drone flights over the project site to collect aerial photography, which would allow us to take kind of pseudo NDVI with RGB. Luckily, this coming summer, we will be able to use Western's new multispectral drone, and we'll be able to plan on flying some flights to be able to collect soil adjusted vegetation index, advanced vegetation index, and normalized difference moisture index. These will allow us to track changes in vegetative health, plant density, and plant moisture content. In addition to the drone flights, we will also be with these drone flights, we're also going to try to collect soil or structure for motion point clouds to be able to attempt to track stream morphology changes over time. In addition to this, we will be taking soil moisture measurements to track changes in how long water will be stored in the soil throughout the season. And there is an opportunity for future students to be able to take over this long-term monitoring study and continue that. So progress so far in the summer of 2024, I shadowed and learned from restoration professionals and attended stakeholder meetings. We created a plan for the first stage of the restoration project. Unfortunately, due to permitting delays, we kept having to push back the building of these structures until eventually we had to push it back to the summer of 2025. Instead of doing this, we were able to pivot and work on installation for another project that the BLM was doing higher up in the drainage basin, installing the similar structures. We also added reflective tags to fence lines to be able to prevent the Gunnison sage-grouse from flying into the wires. And we removed a 1/4 mile of derelict fencing with volunteers and backcountry hunters and anglers. So for one of my project deliverables, I'm organizing a Western Student Work Weekend to install some of these structures. This event will hopefully coincide with Julie's class in the fall, Technical Skills in Ecological Restoration. There are several field trips, and hopefully we can line this up with one of them. As part of this work weekend, I was able to get to help with the field work this summer and this work weekend. I got some money from the Haley family to allow us to camp at Joyful Journey Hot Springs. So if anybody is interested in helping out and soaking with a view of the beautiful Sangre de Cristo Mountains, keep an eye out in your email for the dates to be finalized. And then, as you all know, funding for a lot of projects, including this one, was frozen. And so this project was kind of in limbo this coming summer, the completion of this project. Luckily, that funding, we just found out this week, has been released. So this coming summer, we will install the first round of structures for this project, and we'll conduct another set of vegetation studies, drone flights, and soil moisture readings. And then I'd like to thank my mentor, Michael Russell, for all his support and guidance, my project sponsors and funders of my fellowship, Jenny Naring and Carrie Aloya, for providing me with this opportunity to follow around real life restoration professionals and learn from them how to gracefully coordinate a restoration project. I'd also like to thank restoration specialist Sean Connor from Biologic for allowing me to follow him around and learn from his projects. And I want to thank the Fish and Wildlife Service for funding this project, the Employees Program, and the Integrated Public Land Management track. I'd like to thank the Haley family. And I'd also like to thank Dominique Nicardo for helping me connect with this project and for developing this fellowship and for her support along the way. Thank you.
Right on. Well, our first audience question has to do with the charismatic Poncha Pass population. What do you see the role of this population in the species recovery?
Well, I'm not sure exactly. I think the population has been staying pretty steady. I don't know, I'm not exactly sure how it's going to help, but there's such a small amount of them that you want to be able to protect as many of them as you can. I know they tried to import some from other populations to try to bolster this population, and they tagged them, and a lot of them just ended up flying right back to Gunnison. So it was hard to keep them there. But yeah, doing things like this, improving these mesic fields, and will really help. There's quite a bit of sagebrush out there, but they need to make sure to improve habitat of the wet meadows.
And this is slightly more to the actual restoration process itself. Do you have a favorite LTPBR to install? And what do you feel is most effective when you're out there?
Well, it depends on the particular situation. Yeah, every stream channel is different, even within 10 meters of itself. But I think probably one of the most popular structures for people to install is the Zuni bowl, because it's the most fun. But I also like media lunas. They're all fun to do. Really, it's very satisfying to install them and come back even later in that season and see it already working, see more plants starting to grow, and see it being retaining moisture in the soil. So.
Right on. Well, thanks, Patrick. And hopefully you guys now know where to sign up to get to contribute to this process. Give it up for Patrick. All right. So Sophia Reggiani is up next.
Good afternoon, everyone. I'm Sophia Reggiani, and I'm presenting my preliminary results of my MS in Ecology thesis project, treated and untreated sagebrush wet meadows and the insect communities, differences found within these wet meadows, their richness and abundance. A little background, insects are the most abundant organism in any community. They are important for the establishment of a post-restored ecosystem because of the ecological services that they provide as predators, prey, pollinators, and prey items for other trophic levels. They're often used as model indicators for environmental disturbance effects because of their notable presence or absence in that ecosystem, since they are sensitive to ecosystem changes. Pictured here is a very sad wet meadow. You can see the sagebrush in the back and a very incised channel. There has been 38% loss in sagebrush habitat since 2001. That's about 53.8 million acres in 24 years. That's just mind-boggling in my opinion. But with that, there are wet meadows in these sagebrush habitats, and there's been unknown how many have been lost because of the sagebrush loss. And the sagebrush loss has been contributed to things such as overgrazing, agriculture, mining, and off-roading. So as Patrick has explained, there have been restoration efforts to restore these habitats. For the past 12 years, and here in the Gunnison Basin, there have been local, state, private, and federal agencies that have been working to restore these ephemeral wetlands through things, low-tech restoration methods, as Patrick explained, using wicker weaves, one rock dams, sunni bowls, for example. These wetlands are important for to restore or use the sagebrush habitat, specifically to hold, slow, and spread the water flow and rebuild that sediment and vegetation. Notably, it is also important for the federally threatened Gunnison sage-grouse as they use these habitats to rear their young. Hence, with chicks, will seek out the high-quality habitat found within the wet meadows because the chicks, for the first few weeks of their life, will eat insects before converting their diet to forbs. So vegetation structure influences insect abundance and diversity. So with that, vegetation structure and diversity in conjunction with vegetation moisture is going to have a positive influence on insect abundance and diversity. There has been studies of wet meadow restoration vegetation impacts. So there's been a notable difference in vegetation growth in restored wet meadows. However, there is very little information on what insects live in wet meadows to begin with and what their impact has been on the restored, how they have been impacted by the restoration efforts. In conjunction with that, there is a paucity of information on what insects utilize these wet meadows and for these vegetation structures and how they support other trophic levels such as the Gunnison sage-grouse. So my research objectives include 3 things: to compare insect richness and abundance in treated and untreated wet meadows. Treatment means restored and unrestored. I am also comparing community composition differences and trait prevalence in insect communities. So here are my study sites. I like to describe it to people of like where geographically this is, right behind the landfill and behind the uranium tailing mine that they buried there. Really nice place. So this is Sage Hen and Chance Gulch. We chose this area because they are one of the first restored sites to be conducted. So they're at the time of this research, 12 years old, now going into their 13th year, and also for climate and geographical continuity. I have 5 treated sites and 5 untreated sites, as you can see there. And for orientation, site 6 is like uphill, and we're going downstream as we go north in this picture. So at each site, I have a malaise trap that I designed. Usually the malaise traps that you will see online are the size of a human tent. And I needed that to be much smaller. So malaise traps were made out of bamboo sticks with some no-see-um mesh. And they're angled upwards because for insects, they think up means out. So they're funneled to the top of it and then dropped into a little cup right there that's filled with ethanol. These are left out for 1 week at each site. I should also mention these malaise traps are a passive method of sampling. So this is for flying insects primarily. Also at each site are pitfall traps. At each, there are 3 pitfall traps at each site. These are primarily for crawling insects, and these are actually yogurt tubs from City Market. And like your classic cartoon, insects crawling along and they fall in. And there's ethanol, water, and then a drop of dish soap. The rock was put in there so if any vertebrate were to fall in, they can pull themselves out. For year 2, we're going to put chicken wire mesh over the top. Also for these, this was set out for 4 days, and my sample period is throughout the growing season. So that is June through September, and the traps are left out for 1 week at each month. So post-collection, and this is what I'm currently doing right now, is identifying to the lowest taxonomic level, which is usually the family level. In total, I have 78 samples, would have been 80, but 2 got destroyed by an animal. And I am almost done. I still have my September pitfall samples to do, and I'll complete those next week. But so far, I've identified to ambient level. These are some of the insects that I have found. They're pretty cool. And I'm also cataloging the traits, such as size, color, and mobility method. This is for information that we can gather on what insects are utilizing these areas, and their life history traits might render them easier for sage-grouse chicks to eat. I'm also, after identifying, measuring the insect biomass at each site. So here are some preliminary analysis results through R. This is specifically for my malaise traps. So non-metric multidimensional scaling, NMDS, this is a visual representation of the insect communities found at these treatment sites. So as you can see, the axes are unitless, and the distance between points is not a geographical distance, but a distance of similarity. So the closer these 2 points are together, the more similar those insect communities are at each sample. So each dot represents a sample. And the ellipses surrounding these represent a 95% confidence interval that if I were to go out and get another sample in a treated site, it would most likely fall within that circle. This is a visual representation of the insect communities. As you can see, there's a lot of overlap, so there is no difference in this appearance. This appears to be that there is no difference in treatment types. However, when separated by month, there is a difference. We're not looking at treatment type, but solely at each month. You can see how the insect communities are different for the months since this is the temporal changes as vegetation changes as well. From that, their permanova elucidates the differences shown in the non-NMDS, which did find that there is no statistical difference between the treatment types for malaise traps, but there is for over the months. But that again is not by treatment type. And then an ANOVA or Cresco Wallace is analyzed for insect richness and abundance. Again, this is for malaise traps, so there's treatment by treatment type. There is no difference between richness or abundance. However, when separated by month, but not by treatment, there is a difference. As you can see, it follows the seasonality of gunnison. I did do analysis for my June pitfalls, and there was no significant difference between richness, but there was an abundance difference. So it'll be interesting to see if the rest of the pitfalls follow suit. I also will be conducting an indicator species analysis test. This is important for long-term management. So after I'm done with this study, a manager can go out to a site, and if they find this presence or absence of a certain insect, they can say this is indicative of a treatment. So for my malaise traps, the leaf hoppers are, and their presence is indicative of a treated site. And same goes for my June pitfalls. For the grasshoppers and uncommon wasps, such as Cercerini, are indicative of a treated site. So why aren't we seeing really any difference? It could be because of a couple factors. Thank you. First being that the unrestored sites are, though they're unrestored, they're not necessarily the fully incised eroded channels that we're seeing, except for sites 9 and 10, which are downstream. The upstream sites, which are sites 6 and 3, might be receiving the positive benefits of that restoration because of the sedimentation and vegetation buildup. So they're not necessarily incised. They may be drier, but not what we would consider fully unrestored. In addition to that, there are stock ponds that are surrounding this drainage, and that can influence the hydrology and the insects that we could be finding there. Lastly, there's the insect mobility component. These insects can be flying between these sites as well. So for my year 2 this summer, I will be going out to Black Sage Pass, which is by Juanita Hot Spring. And this time my partners and I were specifically looking for the differences between eroded versus treated areas. This research is important for my constituents listed here, but it's also important for our non-human constituents. We need a voice in this. This research more broadly impacts the greater sage-grouse as they have similar life history characteristics. And this is also important for our pollinating species as they're in decline due to climate change and anthropogenic influences. Thank you. I want to thank the Clark School, the Employees Program, my committee, Dr. Susan Washko especially, and Hannah Carroll and Nate Stewart. Yeah, thank you.
Thanks, Sophia. So a couple questions coming in. Somebody loved the drawings. And are there specific invertebrates that the sage-grouse are known or more likely to go after?
Very little literature has been conducted on what sage-grouse chicks eat. They're thinking ants, spiders, beetles. However, more research needs to be done on that. I was thinking like DNA analysis or like feather clippings or something along that. But these are a possibility of insects that are available for their palate.
Gotcha. So not specific to their diet. They don't know that yet. This is talking about some of your trapping methods. Did you assess for interactions between treatment, trap type, and month?
Treatment trap type by month, like
different interactions between, were you getting things more frequently in one form of your traps?
got it.
There you go.
Statistical interactions between treatment types. Not necessarily, I would say.
So it sounds like there's more to be done here. So future MEM students, if you're looking for a project, we need to know a little bit more about what these sage-grouse chicks are eating. Well, thank you, Sophia. Let's give it up for Sophia. So we've got a little bit of time now to open up for questions to our panelists as a whole. So for the group, what was the most difficult part of conducting your research? What were some of the hurdles that you encountered?
Start. Okay. The animal factor, where I was not expecting there to be animals messing with my traps besides cows, but we were told that there weren't any cows on the land, which is true. However, the deer or pronghorn like to mess with them just the same. So that was a little hiccup, but nothing major.
I think for mine, it was mostly the statistical analysis. The way that I had done the count data and my data forms, when I were to put that into statistical analysis, there were a lot of zeros where a species was not found. So finding the right kind of model for that was a little tricky. And I had to ask quite a few people to figure it out. But once that did get figured out, it was smooth sailing.
After I did my initial plant vegetation surveys, I was asked to go back and do some surveys on a different site, but this was in October and the plants were already senesced. So it was really difficult to tell what plants were what. And it would have been helpful to have an undergraduate research assistant of some sort. Doing the plant vegetation surveys by yourself just takes a lot longer than it would with somebody else.
So all of our panelists touched on some aspects of changing climate, human impacts, things that are really changing our landscapes over time. When it comes to restoration, there's often this question of restoring to what? What are some of the benchmarks that you guys contemplate when you think about your projects and their context in the greater restoration field?
A lot of times with restoration efforts like this, you want to find a reference reach, which is, every landscape is a little bit different. So you'd want to find like an area where you're in the nearby vicinity that you're trying to restore things to. So there's always, there's been human interventions for at least 10,000 years in the US. So, you're never really like trying to restore something back to like a natural state. But so it's important to find that reference reach. What are you trying to restore this to? And then go from there.
I think a lot of times when we're looking at restoring for something, we're also looking at the fact that we're losing a lot of biodiversity. And so a lot of our efforts towards restoring is to keep that biodiversity. And I think that in general, that's a good thing. And we should continue towards that path in any way that we can, whether that's herbicide treatments, restoration projects. They all work together to try and keep that biodiversity.
I'm a big proponent of long-term monitoring. It's great to go out and do the restoration, and it might look visually like appealing, but if we just walk away without long-term monitoring afterwards, then we really aren't doing our full extent of the job.
I have a couple more questions. How much time do we have left? Okay, we're gonna get into two more of these at least. What studies do we need to prioritize for the recovery of the Gunnison sage-grouse?
Okay. I think part of what my study would lead into is identifying what are the specific insects that these sage-grouse are eating.
I think along the same note, same thing, like we want to keep them. We want to be able to give them this habitat that is ideal for them, but we also need to understand either they've adapted in a way to what the current climate is and they're, they have other adaptations or they've changed their preferences. We need to look into that as well, just so we can keep up and so that we're not going off of old science. So yeah, monitoring.
Yeah, and I don't know how qualified I am to answer this question, but I would say more like behavioral studies. I know they're kind of an elusive bird and they're hard to tell. So I would think understanding more of what causes them to be disturbed from their normal habits, like their mating habitat. And that's probably the one that's the most understood. But just in general, what types of activities disturb them and keep them from living their life.
I'm going to combine this last question. I'm going to combine two questions that we got. Folks are curious if you're looking to pursue work that follows in line with your MEM projects, and they also want to know what your advice is to future students. So, what'd you learn in your project, and where are you looking to go from here?
Yeah, I definitely like to keep doing this kind of restoration work for work after school's done. I guess some advice for future students is to advocate for yourself. If you're not getting the support or help you need, just ask more people, and you got to be willing to adapt and change, because you go into thinking your project's going to be one thing and it changes. So you have to be willing to do that. So asking for help when you need it.
Yeah, I think that being a part of the employees program too, I've had quite a bit of like science background from undergraduate till now, but including the environmental management side of things. I have the scientific background, but I want to apply it to actual management. So I do think that I'm setting myself up for a future career within environmental management, whether that's government wise or private consulting. And I think another thing that you should really lean on when you're going through these kinds of opportunities through a master's program is to talk to other students, whether that's new students below you or students that just finished doing their master's program. They have a lot of experience now, and they can kind of help you work around mishaps that might happen if you didn't ask.
Yes, so this for my study has really prepared me for what I would like to do post masters, and that is to work within animal behavior in some aspect, and this is sort closely related to that insect behavior in a little bit. And my advice to students is like, be realistic. It's great that you want to like bite off more than you can chew, but also it's okay to rein things back in.
It sounds like for our future students, we are going to look at our scope. We're going to rein it in. We're going to be passionate about what we're following, and we're going to talk to our peers and to our faculty members to get the help that you guys need. Well, this is a fantastic panel. If everyone wants to show a little bit of appreciation to these guys. Thank you. And yeah, appreciate all the hard work that you have put in.